So continuing on with looking at adaptive immunity. Here we have a table that shows us contrasting properties of B cells and T cells. First point, looking at site of maturation. B cells, you find them in the bone marrow, T cells in thymus. Specific surface markers, in the case of B cells, immunoglobulin, in the case of T cells, T cell receptor or several CD molecules. Remember, CD is cluster of differentiation. Circulation in blood, B cells would find low numbers, T cells high numbers. Receptors for antigen, in B cells you find the B cell receptor or immunoglobulin, and in T cells you find T cell receptors. Regarding the distribution in lymphatic organs, B cells you'll find cortex in the follicles, and in the T cells you'll find them in paracortical sites, which are interior to the follicles. Do they require an antigen to be presented with a major histocompatibility complex? B cells, no. T cells, yes. What is the product of antigenic stimulation? In the case of B cells, you get plasma cells and immune cells. In the case of T cells, you get several types of activated T cells, as well as memory cells. General functions, when we look at B cells, we see production of antibodies, which are inactivating, neutralizing, and targeting antigens. Under T cells, cells are activated to help other immune cells suppress or kill abnormal cells, mediate hypersensitivity, and synthesize cytokines. Now, looking at stage 1, which is the development of lymphocyte diversity, the first thing we're going to look at is specific events that are happening in the T cell development. So the maturation of most T cells and the development of their specific receptors is directed by the thymus and its hormones. Other T cells reach full maturity in the GI tract. In addition to the antigen-specific T cell receptor, all the mature T lymphocytes express co-receptors, which are called CD3. CD3 molecules, cluster of differentiation, remember that? CD3 molecules surround the T cell receptor and they assist in binding. T cells also express either a CD4 or a CD8 co-receptor. CD4 is an accessory receptor protein, which is mostly found on T helper cells, and it helps the T cell receptor bind to the MHC class II molecules. CD8, on the other hand, is mostly found on cytotoxic T cells, and it helps bind MHC class I molecules. Like B cells, T cells also constantly circulate between the lymphatic and the general circulatory systems, migrating to specific T cell areas of the lymph nodes and spleen. It's actually been estimated that more than 10 to the power 9 T cells pass between the lymphatic and general circulations per day. Here you see the surface of the T cell. So within the thymus, which is where they mature, and then looking at the surface, you can see here CD3, antigen binding site, T cell receptor, CD4, and MHC1 depicted. Now, under the development of lymphocyte diversity, the next thing we're looking at is the specific events in B cell development. Now, B cells develop in the bone marrow. Just like with T cells, hundreds of millions of distinct B cells develop. These naive lymphocytes circulate through the blood. They are homing to specific sites in the lymph nodes, spleen, and other lymphoid tissue, where they adhere to specific binding molecules. This is where they will come into contact with antigens throughout life. Here you see a B cell surface. Look, you see antigen receptor or an immunoglobulin, also called an antibody, another word that's used, antigen binding site. Here you see an MHC2 marker, here you see an MHC1. Now, building up of immunologic diversity. By the time you have these B and T cells reaching the lymphoid tissue, each one is already equipped to respond to a unique antigen. This amazing specificity is generated by extensive rearrangements of more than 500 different gene segments that code for the antigen receptors in the T and B cells. In time, every possible recombination occurs, so you end up with a huge assortment of lymphocytes. It's estimated that each human produces antibodies with 10 trillion different specificities. While these DNA recombinations allow for the recognition of a huge array of antigens, which is called the repertoire, remember that a single naive or unstimulated lymphocyte can only recognize one antigen with its receptors. Here you see the gene encoding variable regions. These are your sections of DNA. Then you have different RNA transcripts from the same gene, and then you end up with different amino acid sequences in the finished protein. So the gene that's coding for these variable regions has multiple different sections along its length. Because of this DNA recombination, you end up with these very different RNA transcripts from the same original gene. When those transcripts are translated, which is what's happened down here, the resulting protein will have extremely variable amino acid sequences and therefore extremely variable shapes. So T cell antigen receptors are also formed in a similar manner. The specific B cell receptor, which is an immunoglobulin molecule. So in the case of B lymphocytes, the receptor genes that undergo the recombination described are genes coding for immunoglobulin. Immunoglobulins are large glycoprotein molecules that serve as the antigen receptors of B cells. When they are secreted, they are called antibodies. So an immunoglobulin molecule, from a structure perspective, it's a conglomerate of 4 protein chains. 2 of them are called light chains and 2 of them are called heavy chains. The ends of the forks formed by the light and heavy chains contain pockets, which are the antigen binding sites. These sites are highly variable in shape, and so they can fit a wide range of antigens. This extreme versatility is due to variable regions in the antigen binding sites, where amino acid composition is highly varied from one clone of B lymphocytes to another as a result of the gene arrangements, which I just talked about. So those genes can be arranged, rearranged in various different forms. This is what I'm describing again. This is variable region, constant region. Here you have the longer one, the heavy chain, and the short one, the light chain. Okay. So the remainder of the light chain and heavy chain, which you see here, the dark red part, that's the constant region. So that amino acid content does not vary greatly from one antibody to another. It's this part, the variable region, that varies a lot. T cell receptors for antigens belong to the same protein family as the B cell receptors. They're similar to the B cell receptors in being formed by genetic modification. They have variable and constant regions. They're inserted into the membrane, and they have an antigen binding site, which is formed from 2 parallel polypeptide chains. Unlike the B cell receptor, however, the T cell receptor is relatively small, and it is never secreted the way antibodies are. We'll also look at various other receptors and markers that are not antigen specific a little further in this chapter. Clonal deletion and selection. So when we are looking at why or which of the B or T cells is activated by any incoming antigen, in that whole process, that is called clonal selection. After the activation, the B or T cell then multiplies rapidly in a process that's called clonal expansion. So two important features of clonal selection are that lymphocyte specificity is pre-programmed. It exists in the genetic makeup before an antigen has ever entered the tissues. And each genetically distinct lymphocyte expresses only a single specificity and can react only to that chemical epitope. Here you see an image of this. In here, you've got each genetically unique line of lymphocytes arising from an extensive recombination of surface proteins, and it's called a clone. So this stage, you don't need any foreign antigens. Any lymphocytes that develop a specificity for self molecules would be harmful, and they are ending up being eliminated from this pool of cells. That is what is called clonal deletion. So to put this in other words, you could say that a potentially problematic outcome of random genetic assortment could be that development of clones of lymphocytes that are able to react to the cell. Okay? That would lead to severe damage if the immune system is perceiving self molecules as foreign and was mounting a harmful response against the host tissues. So in a healthy condition, these clones are destroyed during development itself, and that would be called clonal deletion. The removal of these potentially harmful clones is the basis of immune tolerance or tolerance of self, tolerance to self. Because humans are exposed to many antigenic substances during their lifetimes, T cells and B cells in the periphery of the body have mechanisms for not reacting to harmless antigens. Some diseases, which are autoimmune diseases, are caused by the loss of this immune tolerance. So the survival of certain forbidden clones or the failure of other systems that comes in is then going to lead to that. Here you see specificity for a single epitope is programmed into the lymphocyte for life. So you have an enormous pool of mature but naive lymphocytes, and they could be differentiated further if there are certain immune stimuli. But when any epitope enters into the immune surveillance system, it's going to encounter specific lymphocytes that are ready to recognize it. That will stimulate activation of that clone, leading to genetic changes, and those will make it differentiate further into an effector cell. Mitotic division, which is basic cell division in any eukaryotic cell, will then expand it into a larger population of lymphocytes. All of them will have the same specificity, and that's called clonal expansion.